Any questions? Okay, it's Friday. I realize you have, after today, 5 more weeks.
That's crazy. Wow.
Crazy. Quiz Monday. I had the right pages up there last time, 251 through 259, but that is only section 3.2. Okay, 3.3 is just a 13, sorry. I haven't had 3 up there. 13.2. I had up there yesterday, I think 13.2 and 13.3. We're only doing 13.2. Yeah. 13.3 is only a little one-page section. Okay. So we're talking about DNA replication, and we spent some of the time on Thursday discussing the fact that, well, Watson and Crick were correct conceptually about how DNA replicated, semi-conservatively, and that was later substantiated by the experiments of Michelson and Stahl, that in reality, DNA replication was much more complicated than Watson and Crick had ever envisioned. And that's the result of several attributes of both DNA and some of the proteins that are involved in DNA replication. So we spent a little bit of time beginning talking about the role that proteins play in replication. And as a little review, remember that because DNA is a double helix, we have to have several proteins involved in the unwinding portion of replication. This establishes what we call a replication fork, where then the 2 strands of DNA can begin to be copied into 2 complementary strands, giving us 2 identical DNA molecules where there was previously 1. That requires an unwinding enzyme, DNA helicase, the topoisomerase enzyme, which is necessary to relieve some of those kinks that form when DNA starts unwinding and yet is still attached up at the other end of the molecule. And then we need those single-stranded binding proteins to temporarily keep the 2 template strands separate long enough for replication to take place. We then also talked about the fact that replication is a very speedy process, and this is partially explained by the fact that there are enzymes involved in the replication process, particularly DNA polymerase. And DNA polymerase is the enzyme that's responsible for basically reading the template strand and then grabbing the proper nucleotide and putting it into position and hooking those together to form the new complementary strand that's being formed from the template. We also talked about the fact that DNA replication is bidirectional and that we can form these little replication forks so that multiple replication bubbles can form on the same DNA strand, as opposed to just doing one of them and having it proceed from one end to the other. And those are factors that help to increase the speed of replication. We talked about the accuracy of DNA replication and how the copying enzyme, the polymerase enzyme, also has built-in editing functions. So as it grabs a nucleotide, if the template says T, then it should grab the A nucleotide and place that across from the T. But what if it grabs the C instead? And then adds another one to that C correctly. So you've got a mistake there. DNA polymerase enzyme can go back and auto-correct those particular mistakes. And consequently, your error rate drops tremendously so that only about 1 every billion nucleotides actually gets incorporated incorrectly. And that's pretty remarkable accuracy. And then we were just at the point where we were starting to talk about some limitations of the DNA polymerase enzyme. And this is what's going to cause kind of the most confusion for you when we start getting into some of these esoteric aspects of DNA replication that are the result of the fact that this enzyme just will only do certain things, and there's some things it can't do. So what's one of the limitations of DNA polymerase? One limitation is that DNA polymerase can synthesize a new strand de novo. What that means is it needs something to attach those nucleotides onto. That enzyme cannot come in and put the first nucleotide into place. And that's a problem. So for example, here is a segment of DNA. This is only one of the 2 strands of DNA. This is one of the 2 template strands. Okay, notice that it's going in the 3' to 5' direction. And so this new strand is going to be synthesized complementary to this strand. So A pairs with T, C with G, etc. But this one is going to be synthesized in this direction, which is the 5' to 3' direction, because these two have to be anti-parallel to one another. Okay? Well, the problem is that DNA polymerase, when it grabs a nucleotide, has to have a hydroxyl group to attach that nucleotide onto. Therefore, how can it put this first nucleotide into place? There's no hydroxyl group there. It's the first one. Okay? So DNA polymerase cannot start new strands when there was nothing there to begin with. So theoretically, replication would never even start. So what's the solution? Well, actually, a different enzyme is used to start replication of strands. And it's not DNA polymerase. It's actually an enzyme called RNA primase. What this enzyme does is it can start strands. But when it starts a strand, it's an RNA enzyme, and it puts RNA nucleotides into the growing DNA strand at the very beginning, rather than DNA nucleotides. That's kind of weird. What's the difference in the nucleotides between DNA and RNA? There's 2 fundamental differences. You know what they are?
There's a different sugar.
There's a different sugar. Okay, in RNA, the sugar is a ribose sugar. RNA stands for ribonucleic acid. And what's the sugar in DNA? Deoxyribose. Okay? DNA. So that's one difference. What's another fundamental difference?
Uracil replaces thymine.
Yeah, there's no thymine nucleotide in RNA. Instead, there is a uracil. Okay? So if we're starting this particular new DNA strand and replicating it, and this is the first nucleotide in the template, an RNA adenine nucleotide would be put in here. And then an RNA cytosine would be joined to it. And then a guanine. What's this next one coming in? Is that a thymine? No, remember this is RNA. So that would be a uracil. So we make this little short stretch of RNA primer first. And then once we've got that primer in place, then DNA polymerase can come in and attach additional DNA nucleotides to this hydroxyl group and grow the chain in this direction. So all DNA replication actually begins with little short strands of RNA as opposed to DNA. Now, does your DNA then have a mix of DNA and RNA in it once it's replicated? Not finally. Eventually what happens is different enzymes are going to come in and cut out these primers and replace them with DNA. But we need the RNA primer first to get the process going. And then the polymerase can elongate that primer by then adding DNA nucleotides onto that. So that's a little wrinkle kind of imposed by this weird limitation of DNA polymerase not being able to start replication on its own. So that one's not too bad. That one's fairly easy to wrap your head around. These primers are only about 5 to 10 nucleotides in length, and then eventually a different form of DNA polymerase 1, comes in and cuts out the primer and patches back in DNA nucleotides. So, here's our template strand. Here is our new strand being formed in its 5' to 3' direction. Like DNA polymerase, RNA primase will only make a new strand growing in its 5' to 3' direction. Therefore, it will continue then to synthesize a new strand. And after a little bit of primer is laid down, then DNA polymerase extends that strand by adding DNA nucleotides onto it. Okay? So the red here indicates RNA nucleotides. The light blue indicates DNA nucleotides. Okay? So eventually the rest of this would be copied. Then we would come back and cut out the primer. And then it would be all DNA. Okay? Okay, here's where it gets a little bit crazy. Another limitation of DNA polymerase. DNA polymerase only works in the 5' to 3' direction. So why is this a problem? Well, it's a problem because DNA is anti-parallel. And one strand runs one direction, one template strand one direction, and the other runs the opposite direction. So this idea that Watson and Crick initially had, that this DNA double helix could just be separated, and then that both strands were made in the same direction as this replication fork continued to unwind the DNA, and that the new DNA molecules were made by both of these strands being copied in this direction. You can't do it, because the enzyme only works in one direction, and the two strands run opposite directions. Well, when people started to figure out that this was a problem, they said, well, maybe we're missing something. Maybe there's another replicating enzyme that is a 3' to 5' enzyme. Okay? That would solve the problem. Okay? Because if this one were going in the 5' to 3' direction, this one's going in 3' to 5' because these are anti-parallel. And so if you had two enzymes, then you could have this model of replication where it untwists and then replication occurs in the same direction on both strands. Never found this enzyme. It doesn't appear to exist. So, how do we get around this? Well, the reality is that it appears that DNA replication occurs a little bit differently on the two DNA strands. And the way that replication occurs on this strand is a little bit different than the way replication occurs on this strand. Unfortunately for you, that makes it a little bit more complicated in wrapping your head around it. The two strands are designated with different names. One of the two DNA strands is designated the leading strand. And the other strand is designated the lagging strand. The leading strand has a form of replication associated with it that we refer to as continuous replication. The lagging strand has a different kind of replication that we call discontinuous replication. And it actually involves the formation of a bunch of little short fragments of DNA initially. And then eventually those DNA fragments, which are about 200 nucleotides in length, have to be stitched together to make an intact DNA strand. These little shorter fragments are called Okazaki fragments in recognition of the person who discovered this. So what's going on here? What's the difference between replication on the leading strand and the lagging strand? Okay, so here's our parental DNA in dark blue. And again, notice that the two parental strands are anti-parallel to one another. So this one runs in the 5' to 3' direction, this one in the 3' to 5' direction. Okay. The leading strand can be replicated in a continuous fashion, meaning it doesn't stop, it's not interrupted. And the key is that this replication fork proceeds in this direction and continues to unwind this DNA. The DNA replication can then just follow along as this molecule is unzipped, and this strand can just continue to be copied as more template is exposed. We do have to have a primer that starts this out, and then once the primer is in place, the DNA polymerase can just copy, 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 copy as more template is exposed. Okay? And it will continue until the replication fork reaches either another replication fork or the end of the molecule. Okay?
So the leading strand of 5' to 3'.
The leading strand copies the 3' to 5' template, but it's being made in its 5' to 3' direction because they're anti-parallel. This anti-parallel business really causes problems.
Since there are different replication bubbles like on a strand of DNA, is it possible that like one strand or like in a different bubble, the bottom strand could be the leading strand and the top strand could be the lagging strand?
Yeah, and if you look at some of the diagrams, that's kind of the case. That's a little bit beyond what I kind of want you to know right now. But yes, that is an implication. Yeah. Okay, now, the other strand, okay, this other template strand, this is its 5' to 3' direction. So its complement that's being formed, its 5' to 3' direction is this way. Okay, opposite what it is over here. So what has to happen is you have to wait a little bit for this replication fork to open up some template. And then as some template is exposed, the RNA primer can come in here. And then once you've got the primer, DNA polymerase can elongate the rest of that template. But then you've got to wait till some more template is exposed for the replication fork to move farther up here. And then another primer has to be formed. And then that's elongated. And that works back toward the previous one that was made. And then we have to unwind some more. And another primer has to start. And then we elongate it. So these individual little fragments that are formed on this lagging strand are the Okazaki fragments. Okay? So for this to all come together and be finished, on this strand we do have to cut out the primer. There's only a single primer, and replace it with DNA nucleotides. Okay? But on this strand we've got a primer for every little segment that was formed. We've got to cut out the primers. We've got to put in DNA nucleotides. And then you've got to stitch together all of these little segments now to make an intact DNA molecule. That requires a different enzyme. We also need, therefore, an enzyme called DNA ligase that will then join the Okazaki fragments together. So, here's a strand, here's the leading strand being replicated. Remember, it's going in its 5' to 3' direction. It's copying a 3' to 5' template. We do the primer, and then we elongate the primer with RNA polymerase. And then as the replication fork proceeds in this direction and we open up more template, we just continuously make and copy this strand of DNA without stopping. Okay? But if we do it in the opposite direction, then we need these Okazaki fragments. So this shows you kind of what's happening at the replication fork, superimposing all of the enzymes, and then showing here the replication bubble, and that shows a single bubble, and there can be multiple bubbles on that same molecule that's being replicated. In reality, this is an oversimplification of what's happening. And actually, all of these enzymes and proteins form a multi-enzyme complex that attaches to the DNA. And in reality, what we think happens is that the DNA molecule is kind of forced through this multi-enzyme complex, and as that happens, DNA replication occurs. And there's a little bit of looping of the DNA that's involved, but this model suggests that the enzymes move along a static or fixed DNA molecule. What we think really happens probably is that this replication complex is fairly static and maybe attached to the nuclear membrane, and then the DNA that's being replicated is kind of pushed through that, and in that process being copied. What I'd like to do on Monday, do you have things you need to talk to them about on Monday?
Maybe for 5 minutes.
Okay, so we got a little bit of introduction. We've got the quiz, obviously. This on Monday, there's several good videos, some that are available from the website, some that are on YouTube. And I think I'd like to show you some of the videos that show you animations of this happening. Because this kind of a static picture, sometimes it's a little bit harder to wrap your head around than an actual animation that shows what's going on. But the key elements are the leading strand only needs a single primer and is done in a continuous fashion as the replication fork moves in this direction. The lagging strand involves discontinuous replication, and each one of these little segments is being formed as that replication fork is opening, and then synthesis occurs away from the replication fork. Okay? Each segment needs a primer to start it out. Eventually we've got to cut those primers out and then stitch these fragments together using DNA ligase to make the intact strand. So ultimately what happens is, when this process is done, this is the daughter molecule, this is the daughter molecule, and the sequence of nucleotides in this daughter molecule is the same as this one, and they're the same that these were to begin with. So DNA is copied and replicated faithfully. Okay?
The discontinuous replication, what exactly does that mean? Does that mean that it is done in fragments, or does
Yeah, it's done in fragments, and you really kind of have to wait for replication to happen until the fork opens up some template. So a little bit of replication happens and it stops. And then more template is exposed and then it starts up again. Okay? Whereas in continuous, since it's moving toward the replication fork, you just kind of keep moving along like a train going down the track. Okay? Here the train's going down the track the opposite direction. Okay? Much of this understanding of DNA replication was originally worked out by doing studies of bacterial DNA replication, particularly E. coli. E. coli have a circular chromosome. There is only one chromosome in E. coli. Eukaryotic replication, we think, happens in much the same fashion, but eukaryotic chromosomes are much more complicated than prokaryotic chromosomes are. They've got a lot more protein associated with them, and the chromosomes are all folded up into these fairly elaborate structures that we call chromosomes. And one of the things I'll show you at the end, if we have time, is a picture that kind of shows you the relationship between a DNA molecule and then a chromosome and how they're related to one another. So here are some of the enzymes that are involved. This would be a good slide to use for studying for the exam or for the quiz, for that matter. And talks about the enzymes that are involved in the synthesis of both the leading strand and the lagging strand. Okay. We've talked about the fact that DNA replication is pretty accurate. And even mistakes that are made during replication are usually auto-corrected. Well, that's really important, okay? Because if you get a permanent change in a DNA molecule, it could be a result of an error of replication. What do we call that? It's a mutation. And what are the consequences of having a mutation?
Cause a change in your gene expression.
Okay? A change in your genes can actually make a change in the proteins that are produced. And if you produce a different protein, a wrong protein, a malfunctioning protein, can that have an effect on your physiology? Absolutely. Absolutely. We tend to think of mutations as being all bad. Some mutations are fairly neutral. They're there, but they don't really affect the three-dimensional structure of the protein. And remember, it's the three-dimensional structure of the protein that determines its function. But some mutations are actually beneficial. You guys spent a fair amount of time last semester talking about evolution, didn't you? And the importance of genetic variation as a raw material for evolution. Where does a lot of that genetic variation result from? This random mutation. In one of the labs later on, you're going to see a video called Accidents of Creation. And it's going to talk about the role that mutation has played in the evolution of plants and animals, and spends a fair amount of time talking about the evolution of a group of animals, which is a direct result of a single mutation. That group of animals are called ruminants. Anybody know what ruminants are?
Animals with 4 stomachs.
Animals with extra stomachs that can feed on grass and digest cellulose. Okay? And the evolution of ruminants, as it turns out, was an important selective force in the evolution of a new type of plant called grasses. And so a single mutation is, we believe, responsible for the evolution of ruminants and the evolution of grasses. Well, most mutations are harmful. And one of the things that we've come to understand about mutation is that mutation is typically one of the very first events that happens in the development of a cell becoming cancerous. In actuality, for a cell to become cancerous, there probably are multiple mutations that have to take place, and we'll talk about what some of those are later in the semester. Even if DNA has been replicated perfectly in your cells when they divide, there are environmental factors that can cause damage to your DNA and cause mutations. And some of the environmental factors include various chemicals as well as ultraviolet radiation. That's why public health experts are becoming so vociferous about trying to warn people about the danger of overexposure to sunlight and particularly tanning beds, because they contain ultraviolet radiation. And ultraviolet radiation can damage DNA. Here's how they do it. Oops. This one's a little bit hard to see. So here's a normal segment of DNA. 2 strands, A pairs with T, G pairs with C. And in order for replication of this DNA to occur, these 2 strands have to separate, and then replication occurs. Okay? Ultraviolet light can be absorbed by DNA. And particularly the thymine nucleotides in DNA are affected by ultraviolet light. And they start to form, in response to ultraviolet light, some abnormal bonds that weren't there before. Okay? There wasn't a bond connecting these 2 thymines directly to one another, a covalent bond. Here there is. This shows you what that covalent bond looks like. This particular new bond is called a thymine-thymine dimer. And a thymine-thymine dimer distorts the shape of the DNA. So this would be a segment of DNA that had a thymine-thymine dimer. When this altered DNA tries to replicate, it can't. That thymine dimer interferes with replication. And that's a problem. So, there are some repair systems in cells that are able to detect the presence of these distortions in the DNA, cut those segments out, and then fill them back in with the proper nucleotides, because we've got a template down here to do that, and basically to repair that particular damage that's occurred. That particular type of repair is called excision repair. And one of the key enzymes that's involved in that is an enzyme called a nuclease. So the nuclease chops out these damaged segments, and then a DNA polymerase repair enzyme can come back in and fix in the hole, basically, and repair the DNA to its normal state, where it can replicate again. Okay? So repair depends upon properly functioning enzymes. You've got to have a properly functioning nuclease. You've got to have a properly functioning DNA polymerase repair enzyme. Okay? Sometimes people have genetic defects in those repair enzymes. And if they do, then these distortions and damages to DNA aren't fixed. And that can result in mutation, and that can result in cancer. There is a genetic disease called xeroderma pigmentosum. And individuals who suffer from this particular genetic disease have one or more defects in their repair mechanisms for DNA repair. And consequently, these individuals are very, very prone to getting cancer from exposure to sunlight. So even exposure that would be normally kind of tolerated by the average person, because you have repair enzymes to deal with any damage that might occur, these individuals suffer consequence from. So if they go outside, they've got to heavily cover themselves with protective clothing, use, super sunscreen. It is a really serious condition, all a result of a defect in some of these repair enzymes. So correcting damage that occurs to your DNA is really an important part of your body's homeostatic process. Now, we're going to come back after we talk about gene expression and learn a little bit more about the genetic code and transcription and translation, and talk a little bit more about mutation and some of the types of mutations that can occur and some of the consequences that can occur as a result of mutation. Okay, so we began our discussion of DNA talking about why people were interested in chromosomes. Okay? This represents a metaphase chromosome. Y'all remember what metaphase is? Okay, remember in metaphase, what's happened is, remember in interphase, the DNA is in this kind of tangled, elongated state. And then that material starts to shrink or condense and to form visible chromosomes. And then the chromosomes will eventually line up on the spindle fibers on the metaphase plate, the equator of the cell, and then be pulled apart and apportioned to the two different cells that are dividing. So this would represent a metaphase chromosome. Is this the same as a DNA double helix? No, it's not. And most people have that misconception that a chromosome is equal to just a DNA double helix. This is not a metaphase chromosome. Okay? So what's the relationship? Well, I'm just going to show you right now for kind of contextual information, and we'll come back and dig into this a little bit more when we talk about regulation of gene expression. So, here's our DNA double helix. Okay, to look familiar. Two strands. The diameter, and don't worry about these numbers, of a DNA double helix is about 2 nanometers in diameter. It's pretty thin. Okay. DNA has to become massively folded up to form a metaphase chromosome. It's got to be coiled and twisted back on itself and twisted around other proteins before you form this complicated structure. So this contains all kinds of DNA double helices, all twisted, wrapped around one another in actually very organized ways. One of the proteins that's really important in folding up the DNA to make a chromosome is a type of protein called a histone. And we'll come back and talk about histones a little bit later on. But basically, these histones associate with DNA, and DNA double helices twist around a group of histone molecules and form kind of a little loop structure. Okay. These little loop structures are called nucleosomes. Sometimes they're referred to as beads on a string. So the nucleosome is the bead, and then the DNA that isn't twisted up is the string. We form all these beads on a string. Okay? The diameter of this is about 10 nanometers. Okay? That's bigger than the double helix. Okay? Well, then nucleosomes can all twist up around one another to form a more complicated, wider structure called the 30 nanometer fiber. Okay? So this is a bunch of nucleosomes all wrapped up around one another in a very organized fashion. The 30 nanometer fiber can then be twisted into a loop structure. So all of these 30 nanometer fibers, this is one 30 nanometer fiber, all become looped around one another to form this 300 nanometer fiber. And then these 300 nanometer fibers twist up around one another to form a metaphase chromosome, which is about 700 nanometers in width. Okay? So don't get all hung up right now about the numbers, 30 nanometers, 100 nanometers, whatever. But just understand that DNA is successively folded up in more complex fashions, usually involving histone proteins facilitating that folding before you form a metaphase chromosome. Now, one of the next topics that we're going to talk about is gene expression and how the instructions of DNA get turned into protein. Well, in order for gene expression to occur, what we have to do is we have to copy DNA into RNA. That process is called transcription. And then the RNA has to be translated into a protein. So basically, there's a whole bunch of new enzymes that are involved in these processes. And if those enzymes can't get to the DNA to copy it, we're not going to have gene expression. Those proteins aren't going to be made. So this is pretty much transcriptionally inactive. Okay? So is this. This is even pretty much transcriptionally inactive. The enzymes really can't get to the DNA to copy it. So, just like this. It's really when you get kind of down to this point where the DNA becomes pretty much exposed as a double helix, the transcription can actually occur. So you got this really interesting dynamic in the cell. At some points of the cell's life, during the M phase of the cell cycle, the chromosomes look like this. In interphase, the chromosomes look more like this or this. They're not quite as folded, but they're still somewhat folded. But for transcription to occur, we got to get back to kind of this in order for enzymes to have access to the templates to copy them. So there's this real dynamic that occurs within the cell of DNA folding and unfolding, and that process really has to be regulated if we're going to be able to regulate how genes are transcribed. What's your question?
You kind of answered it. So basically, in interface, they're just sort of twisted on those stones.
Yeah. In the interphase, it's kind of in this somewhere in this ballpark. Okay. So some of the DNA is accessible for transcription, others isn't, and sometimes we've got to unfold these nucleosomes to transcribe these genes during interphase. Okay. Okay, so now that you understand, hopefully, in a little bit more detail, something about the structure of DNA and how it's replicated, we're now ready to attack one of the more important concepts in biology and the cornerstone of molecular biology, the central dogma. What is a dogma? A dogma is a belief. Okay? So this is a central belief to biology. Does anybody know what the central dogma says?
DNA to RNA to protein to trait.
DNA, RNA to protein. The central dogma describes the relationship between genes and protein. Or, if you like, genotype and phenotype. What's this process called? DNA to RNA. That is transcription. And in eukaryotic cells, in your cells, where does transcription take place? In the nucleus, okay? What's this process called? RNA to protein. That's translation. And where does that happen? Happens in the cytoplasm. And what cell structure has to be present for translation to occur? What's the machinery in the cell that makes proteins? Ribosomes. Okay? So this is going to happen somewhere in the cytoplasm. Those ribosomes might be associated with membranes, so they might be membrane bound. But they might be free ribosomes as well. Depends on the kind of protein that they're going to be making. Well, the central dogma introduces some kind of interesting concepts and some things you need to start kind of thinking about. And one of them has to do with kind of molecular language. What do I mean by that? Okay. What is the language of proteins? Amino acids. Okay. The way you make a protein is you string a bunch of amino acids together. Where do those amino acids come from? Can't make protein without them. Where do they come from?
You mean for autotroph or heterotroph?
Either. Where do they come from in your cells? Where do your amino acids come from in your cells that you use to make new protein?
Food.
From food, right? So some of them you ingest directly, and your cells do have the capability to kind of convert some amino acids into other ones. Okay? Ones your cells can't make and you've got to get in your diet, do you know what those are called? Those are called essential amino acids. Okay? They've got to be present in your diet. Other amino acids can be made from other ones. Okay? Doesn't mean they're not important, but they're referred to as non-essential amino acids. What's the language of DNA? Nucleotides. What does DNA represent? It represents information or instructions. And what kind of information does it represent, particularly relating to proteins? DNA represents the information for the primary structure of a polypeptide chain. Remember what primary structure is? What is it?
It's a chain of amino acids.
It is the kind and order of amino acids in a polypeptide chain. So if these letters of the alphabet represent amino acids, are these the same primary structure? Got the same kind of amino acids, don't they? But they're different orders. What does primary structure dictate in a protein? Does it dictate secondary structure? Does it dictate tertiary structure? Does it dictate function? Yep. So this potentially could fold up a different way and make a different functioning protein than this does. Well, how does the cell know which amino acids to string together in the cytoplasm, which ones it needs to put together, and in what order? That's encoded into the instructions of DNA. Okay. But how do we get this language problem resolved? The encoding information is in the form of nucleotides, A, T's, G's, and C's. But the protein is amino acids. Threonine, lysine, isoleucine, phenylalanine. Okay? So how does a bunch of A, T's, G's, and C's provide the genetic information for sequences of amino acids? That's what we need to dig into. But this is what the central dogma says. If there's one big picture thing that you can take out of this course, or any biology course, I would like you to really understand that ideas in biology are always subject to change, and typically any rule in biology has exceptions. And the central dogma is something that has exceptions. This particular idea represents the idea of a one-way flow of genetic information, that DNA is always converted into RNA, and then RNA is always copied into a protein. We've now found exceptions to the central dogma, particularly situations where RNA can actually be copied into DNA. That particular process is called reverse transcription. And requires an enzyme called reverse transcriptase. In molecular biology, many times when you see exceptions to rules, oftentimes the exception is a virus. Viruses are weird things. And one of the virus that can do this reverse transcription, that's called a retrovirus or a reverse virus. What's the most probably infamous retrovirus? Something that's got RNA and converted into DNA. HIV. Okay. We'll talk a little bit about HIV as we kind of dig through this central dogma. So where we're going to start on Tuesday will be beginning a discussion of genes and what a gene is, and how the genetic code is organized, how those A, T's, G's, and C's provide the coding information for making a protein. Okay? So don't forget Thursday lab, look at your plates.